Hello, my name is Erick Armingol. I'm a PhD candidate at the University of California, San Diego. I'm working in bioinformatics and system biology. And today I'm gonna present our poster about unraveling the complex cell-cell interactions driving phenotype with tensor cell-to-cell. So cell-cell interactions and communications are usually shaped by the cellular context where they are happening. This context could be, for example, different diseases that an organism could have, different life stages where the organism is at the moment, even the different tissue microenvironments where the cells are located. Cell-cell interactions and communications are mediated by ligands and receptors, and these are usually used as signals so cells can coordinate multicellular functions, and this is associated with the phenotype of an organism. To measure and have an idea of what ligand and receptors cells are using, we can use single-cell technologies, single-cell RNA-seq. And with computational tools, we can get the gene levels of the ligand and the receptor and infer how cells are using them. But when we are interested in studying the cellular context and how they shape the cell-cell communication, as I mentioned, could be the different life stage, disease condition, or even a location where the cells are. Existing tools are very limited in that sense, because what they do is usually they infer the cell-cell communication for a given context. That could be a sample, and they perform that for each of the contexts or samples we have. And they, at the end, they perform pairwise comparisons between two contexts. So we can have an idea of the changes that communication could have from one condition to another, but we only summarize this information in this pairwise comparison. But if we're interested in having the idea of the dynamics that cell-cell communication follows across multiple contexts, we would lose a lot of information about the correlation structure in the data with the phenotype we are interested. So there is a need for tools that consider all the contexts simultaneously without losing information, and in order to have an idea of the dynamics of cell-cell communication. In this work, we propose tensor cell-to-cell, a method based on tensor decomposition that is able to extract changes of cell-cell communication across multiple contexts. A tensor basically is an multidimensional organization of data. The one that we usually know is a matrix organization that corresponds to a tensor of 2 dimensions. But we can have data with multiple more dimensions, as for example, a picture. A picture contains multiple pixels, and each pixel corresponds to a vector of the level of colors that the picture have in that given pixel. So we can arrange this information as a 3-dimensional tensor, with the first 2 axes corresponding to the X and Y axis. So we have the pixel, and the 3rd dimension is the channel, the color channels that each pixel have, and these could indicate the level of reds, greens, and blues, for example. So when we run tensor decomposition of this tensor, what we're trying to do is to extract and summarize the original tensor into a compressed tensor that correspond to, in this case, to the sum of multiple factors, and each factor would represent the most prominent parts of the original tensor. In this case of the picture, we can extract, for example, the pillars in this sphere, some intensities related to the light at different location of the picture, and some reflection of light on the water, for example. Following this idea, when studying cell communication, we can arrange all the inferred information for each of the contexts, put them together into a factor, and try to identify the most important parts of this communication, summarizing multiple cells and ligand receptor pairs that could be used depending on the context where they are occurring. Tensor cell-to-cell, what basically does is We have the database of ligand receptor pairs, the gene expression level, and for each ligand receptor pair, we build a communication matrix with all the pair of cells, the one sending the ligands and the one receiving those ligands through the receptors, and we can assign a score for each of these pairs. We repeat that for each ligand receptor pair to build a three-dimensional tensor with this information like sender cells in one dimension, receiver cells in the other, and ligand receptor pairs in the third. So we keep the correlation structure in this data. And then we can repeat that for each of the different contexts we are interested in to build a four-dimensional tensor. So tensor cell to cell run a tensor decomposition on this four-dimensional tensor. To get multiple factors, we have to specify a number of factors. And each of these factors would capture a prominent part of the cell-cell communication processes across all the contexts. These factors could be summarized by vectors, one vector for each of the dimensions in the tensor. So we can get loadings for the context in a given factor, the ligand receptor pairs in that same factor, and which one are the senders and receiver cells. So we can have the dynamics of the communication across contexts. For example, if they are time points, here we would observe that they are decreasing across time points, identify the most important ligand receptor pairs and the cells producing the ligands and the cells receiving those ligands. So to test our approach, we simulated a system of 3 cell types using 300 ligand receptor pairs. For multiple groups of these cell types sending signal to other cells or to themselves, we assign a given number of ligand receptor pairs, and we simulated 4 different patterns, and we included them in this 4-dimensional tensor that we simulated. Tensor cell to cell, without knowing anything about this simulated information, just the numbers in the tensor, was able to decompose this original tensor into 4 factors. And if we see the loadings for the context dimension, they follow the same patterns that we simulated before performing the decomposition. And in this context, tensor cell to cell capture the important ligand receptor pairs, the one we are assigning a given pattern, and the cells involved in this pattern as well. So with this idea, we were able to test real data. One case was associated with COVID-19. So we have patients with multiple different severities of COVID-19. We generated this tensor for the sample from bronchoalveolar lavage fluid. And this includes immune cells and epithelial cells as well. We got around 10 factors, but here I'm showing just 5 of them. And in the first 2, we observed that there is a correlation between the loadings that we got and the severity of the patient of each of these loadings. And this seems associated with communication between epithelial cells and immune cells. And when we look into the top ligand receptor pairs of these factors, they are associated with the recruitment of immune cells. Other factors distinguish the severities, moderate versus severe case, for example, factor 3 and 8, and factor 9 seems to capture communication that is occurring mostly in control samples. The main difference between factor 3 and 8 is at the macrophages level that are statistically significant. And when we look into the molecules, the factor associated with severe cases seems to be pro-inflammatory macrophages, while the one associated with moderate cases is anti-inflammatory macrophages. The other data set is about brain cortex samples of patients with autism spectrum disorder. So we have an idea of how neurons communicate with non-neuronal cells in the cortex. And we obtained 6 factors where 2 of them were statistically different at the level of patient groups, the ASD condition versus the control one, and mainly involving neurons at the different cortical layers, and the other one inhibitory interneurons. So in contrast to the COVID-19 case, we can use the context loadings also to try to cluster the patients, given the combination of self-communication capturing the different factors. And here, we were able to have together a patient with ASD condition versus control, and we can observe the dysregulation of self-communication given the factor that seems to be associated with one condition versus the other. Also, instead of having the top ligand receptor pairs, we can perform analysis by a gene set enrichment analysis using the loadings of the ligand receptor pair in each of the factors. So we can perform the analysis in a factor-specific way and obtain what biological processes could be involved in each of the factors. And here we were able to observe, for example, factor 3 and 4 that seems to be associated with the control condition versus the ASD, or increase at least in the control condition. Seems to be associated with axon guidance, some cell adhesion molecules in some case, and the ERBB signaling pathway. So with this, tensor cell-to-cell seems to be a good approach to study cell-cell communication across multiple contexts that works for both simulated data in this case and the real data we evaluated, which is consistent with previously reported articles at the level of mediator we detected. And these details about molecular and cellular participants could be potentially useful for therapeutical target identification, in other words. So if you have questions, you can contact me with this information here at the end. You can download tensor cell-to-cell and use it. And if you like, you can read the full article about the methodology and the example we provided. Thank you for your attention.